In this video, we will discuss about TAM, that is tumor-associated macrophages targeted therapies, and these therapies are targeted in order to cure the cancers. So let us first understand what are these TAM or tumor-associated macrophages. The TAM has these following properties, and TAM are that is tumor-associated macrophages are differentiated from the monocyte, which is a part of the innate immune system. So in our previous videos we have discussed whenever the in our last videos or previous videos we have discussed that whenever our system observe any kind of infection, it will recruit monocyte, and the monocyte will as it is a part of the innate immune system, it will try to kill those infection area or the foreign bodies that are harmful to the host. However, in the case of cancer, what happens is that it will differentiate this monocyte to this tumor-associated macrophages, and that can help the cancer cells in immune evasion, and also helps the cancer cells to proliferate. So what are the ways that it can help the cancer cells to proliferate? Is that it can help in this angiogenesis, that helps in providing the nutrient supply that is essential for the uncontrolled growth rate of the cancerous cells, that can be seen here. And apart from this TAM also helps in the metastasis. So metastasis is the process by which tumor cells spread from one part of the body to another part of the body. And how they helps in the immune evasion is that through these checkpoint ligands that are overly expressed on tumor-associated macrophages, such as this PDL1, B7H4. So it will interact with the T cells or NK cells to suppress the immune response against the tumors, and simultaneously it will also recruit this Treg or T regulatory cells that will also suppress the immune response against the tumor cells. So this is how the monocyte was main purpose is to deal with the infection, will end up differentiating into TAM, that is tumor-associated macrophages, that helps in the proliferation of the tumor cells. Uh, so how this TAM can now be now there are various strategies trying to develop that can use this TAM in order to deal with the tumors or solid tumors. So some of the strategies are shown in this panel B. So one of the one of the strategies is this immune checkpoint blockade, where uh, this checkpoint that are present on these tumor-associated macrophages, such as this PD1, SIRPA, Siglec receptors, LIR, RB, could be inhibited artificially, could be inhibited by these checkpoint inhibitors, so that the immune system will not be suppressed by these tumor-associated macrophages. Other things that they can do is they can load this nanoparticle with this monocyte, so that it cannot be differentiated themselves towards this tumor-associated macrophages, uh, and act and then on the way it can act as it can differentiate itself into from monocyte to the dendritic cells that can act against the tumor cells, or that can have the anti-tumor properties. Other strategy that that can be used is derived from the T cells. That is, instead of modifying T cells into the CAR T cells, they can one can modify these monocytes into these CAR M cells or CAR monocytes, so that these monocytes can then be used to detect the antigens that are found on the tumor cells, and can trigger the immune response, that as can be seen in this figure at the bottom of this figure, that is highlighted by the laser right now. And other things that can be done is this cytokine expressing monocyte. The monocyte can be engineered in a way that it will express cytokines that will trigger the immune response to act against uh, to act against the to act whenever these cytokines are released, and these cytokines will release whenever they interact with the tumor cells. So in this way, there is cytokine expressing monocytes can be engineered. And apart from this, other ways are those that whenever the monocyte is differentiated into this TAM, this tumor-associated macrophages, it will also generate immune suppressor agent. So these immune suppressor agents can be reduced by inhibiting the function of this CSF1, CCR2, CCL2, so that whenever this TAM are generated, it will not have the immune suppressing agent, so that it will not suppress the other immune response that can arise on that can act on these solid tumors whenever they are detected. The other idea that is mentioned here is about this TAM activation. That is, TAM can be activated to act against the solid tumors by inhibiting this kind of receptors, that is CD40, TLR, or STING. So these receptors are being interacted by the solid tumors or tumors, so that this TAM will not act against those solid tumors. So when the idea is that whenever these receptors are being blocked by some other antibodies, uh, so that the tumors will not be able to suppress this TAM or these macrophage functions, and this macrophage can do its original function, that is to act against uh, the foreign bodies that are harmful to the host. Other things that can be done is to block the metabolic regulators, such as this glycolysis, that is essential for this glycolysis and idea that is essential for the growth of this TAM. So by blocking those metabolic regulators, this TAM will not be able to survive. Uh, TAM will not be able to survive for a long time, and thereby this TM will not do the immune suppressor functions that it usually does whenever it is differentiated by the tumor cells. Whenever the monocytes are differentiated by the tumor cells and converted into the tumor-associated macrophages. So these are some of the TAM targeted therapies that can be used to improve the immune response against the cancerous cells or solid tumor cancerous cells. So I hope you got an idea how these TAM targeted therapies uh, are being are going under uh, research, or how these TAM targeted therapies can be used in future against against the diagnosis of the solid tumor cancers. If you need to understand more about this STEM targeted therapies, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.